Okay, our third PowerPoint in Unit 7, Make a Carbon Copy. Our quote from the French philosopher Jean Baudrillard, whose work was the inspiration for the Matrix film series. The very definition of the real becomes that of which it is possible to give an equivalent reproduction. The real is not only what can be reproduced, but that which is always already reproduced, the hyperreal. Our learning goals for this presentation: be able to differentiate between binary fission and mitosis, which we learned about in the last PowerPoint, and our new vocabulary word, meiosis, and then be able to outline the advantages and disadvantages of meiosis. We already know that DNA replicates itself through a mechanistic process, meaning that there's a very specific mechanism it goes through. It's very methodical. We know that bacteria make more bacteria through binary fission, and we know that eukaryotes make more eukaryotes through mitosis. What we learned in the lab is that both mitosis and meiosis are a series of steps that follow a pattern with many of the same steps. And also, do anyone actually clean pipes with pipe cleaners? So, somatic cells, from the Greek soma, meaning body. So almost all of our cells are somatic cells, and they get specialized to different tasks. Like we have our cells that are in our brain use different proteins and do different things than the kind of cells that are in our skeletal muscle versus our liver, but they're all somatic cells. They all have the same DNA, but different parts of it are active, doing different things. So each one of them has a certain number of chromosomes, that tightly wound DNA that we talked about. They always show up in pairs, homologous pairs. Pairs because we get one from the mother and one from the father. Humans, our somatic cells, have 46 chromosomes. If you line them all up, we call that a karyotype. That's the images at the very bottom. And we just numbered them in order of size. So chromosome 1 is the largest, and chromosome 22 is the smallest. The 23rd pair are the ones that get kind of mismatched, because you can either have two X chromosomes or an X and a Y chromosome. Those are our sex chromosomes. Now, every species has a different number of chromosomes. I just said that humans have 46. But chimpanzees have 48, and in fact potatoes have 48. Chickens have 78, but frogs only 26. Peas 14. You see the numbers are all over the place. There's another kind of cell. So most of our body is made out of somatic cells, but the other kind of cell is a germ cell. It's found in the reproductive organs like our ovaries and testes, and they develop into gametes. Gametes are the sex cells. So that means we're talking about the gamete that's called an egg and the gamete that's called a sperm. Now, gametes only have half as many chromosomes as somatic cells. Because each gamete's only bringing one chromosome with the pair. So together, when you get a sperm and an egg, they'll fuse to form a cell that has the full set. So in the picture we have, in situation A, we have an egg that has 22 autosomal chromosomes plus an X sex chromosome. So it has 23. The sperm is also carrying 23 chromosomes, 22 of the autosomal chromosomes and then one sex chromosome. Together, 44 chromosomes plus 2 sex chromosomes, 46 total chromosomes. In B, we see the same thing, except the sperm has a Y instead of an X. But still, egg plus sperm, you get half from one, half from the other, one full set. So we call that first somatic cell a zygote. That comes from the Greek zygotos, meaning to join. It's the first somatic cell. So the gametes we refer to as haploids, and the somatic cell as a diploid, from Greek haplos, meaning single, and diplos, meaning double. So just like single plus single is a double, a haploid plus a haploid is a diploid, meaning you take one gamete and add it to another gamete, and you end up with a zygote, a somatic cell. Developing question: what would happen to the zygote if the gamete was diploidal? Well, if each gamete was a diploid, meaning it had, in the case of humans, 46 chromosomes, then the zygote would have 46 plus 46. So it'd have, 92 chromosomes? It would not develop effectively. It probably wouldn't even live. It's too great of a genetic abnormality. Well, let's continue to imagine this make-believe scenario that somehow that zygote not only lived but reproduced, and it happened again. And let's say 5 generations later. So if the gametes keep giving the full set of chromosomes, what happens? Well, that means each daughter is going to have more chromosomes in their cell than the one before. I mean, eventually, the cells is going to be all filled up with chromosomes. It's going to be a big problem. Not going to work. What does a species' quantity of chromosomes imply about the species itself? Does the number of chromosomes you have mean something? Here's an easy one. Nothing. It doesn't mean anything. A fern has almost 1,200, whereas a fruit fly has 8. The fact that humans have 46 and some things have more chromosomes than us and some have less says nothing about us as a species or anything else. Here's a mastery question. Would you predict the chromosomes of a bacteria to be haploidal or diploidal? Justify your answer using clear. Well, the claim I'm making is that bacteria have a haploid. My evidence is that they reproduce through binary fission. My reasoning is, since there's no sexual reproduction, no gametes, they're not going to get a pair of anything because they don't have parents. So there'll be no diploid because you're not adding 2 sets of chromosomes together. Therefore, bacteria have merely a haploid. The process of meiosis is a lot like the process of mitosis, which is why they're so similar. The osis part comes from the exact same word. It's a process. Now, mitosis, remember, we use the Greek mitos, meaning thread, that Fleming saw. Here, a German scientist who lived right around the same time, just a little bit later, used the Greek word meion, meaning less. So the Greek word just happened to come out to be very similar. So meiosis is a lessening process in the cell. It's how a diploid cell makes haploid cells. So after prophase, the DNA gets wound up and the 2 sister chromatids, one from the mother, one from the father, gets attached by the centromere. We call those a sister chromatid. Now, your chromosome that you get from your mother and from your father are homologous chromosomes. Whenever you go through DNA replication, the chromatid you got from your mother gets duplicated, and those 2 get joined together to form a chromosome. The chromatid you got from your father gets duplicated, and those 2 get joined together to form a chromosome. So the replicated part is what we call the sister chromatid. It's the exact replication. The one from the mother and the one from the father, those are the ones that are called homologous chromosomes because they're the same kind of chromosomes, but they have different information on them. So let's start with the similarities. Meiosis still starts with interphase. We still have G1, S, and G2. That's growth, synthesis, growth. We still go through a lot of the same process, the PMAT, prophase, metaphase, anaphase, telophase. In prophase, the nucleus breaks down, the chromosomes pair up. In metaphase, as homologous chromosomes line up down the middle. In anaphase, they're pulled apart at their centromeres and they're heading in different directions. Telophase, the two new nuclei are forming and the cell splits in two, each of them having 23 pairs of homologous chromosomes for a total of 46, just like mitosis. But then, something completely different. After telophase happens in meiosis, we go off towards a second prophase. Now we don't have interphase again because we're not making any more DNA. So in prophase 2, they get all dissolved out of the nucleus. In metaphase 2, whenever we line up, we're only lining up 23 chromosomes. In mitosis, we line up 23 pairs of chromosomes. We're not lining up pairs of chromosomes anymore, just 23. Now, each of those 23 still has its homologous chromosome, but because we did not go through replication, we do not have any sister chromatids. So now in anaphase, instead of pulling it so that a homologous chromosome goes to each side, now we're pulling it so that only one of each sister chromatid goes to each side. Telophase, the second telophase happens similar to the first one. The cell separates, new nucleus forms, but now we're ending with haploid cells. So after telophase 1, we had 2 diploid cells, and if that was mitosis, it would finish cytokinesis and that'd be it. In meiosis, telophase 1 ends with 2 diploid cells, and then telophase 2 ends with 4 haploid cells, 2 each. So meiosis is a longer process. So we start in interphase, it's normal. We have our 2N. N refers to the haploid. So in humans, N equals 23. So a diploid cell, a somatic cell, has 46. So in interphase, we have our 46. And then we go through the S part of interphase where DNA replication happens. And now in prophase 1, we have 4N. We have double. We have 2 sets of our DNA. In metaphase 1, the chromosomes are lining up the center. We still have 4N worth of DNA. Anaphase 1, chromosomes are being pulled apart. Telophase 1 in cytokinesis separates the cells entirely. Prophase 2, both cells have 2N. Metaphase 2, chromosomes lining up. Both cells still have one full set of DNA. Anaphase 2, that full set starts to get pulled apart. Telophase 2, that starts to get separated until we're left with 4 granddaughter cells, which each only have N, a haploid. So gametogenesis is the genesis of gametes. It's how we create gametes. It's how we create sperm. It's how we create eggs. It uses meiosis to form haploid cells. Now, this is slightly different whether we're doing the creation of a sperm cell, the creation of an egg cell. Whenever we create a sperm cell, first of all, sperm must be very motile. So we need the formation of a flagellum, that movement. That means some new proteins need to be synthesized. It also means the cell is reshaped to be streamlined. It also means that really all the sperm needs to contribute is the DNA. The other parts are only useful for the motility. So we need to move, we need to get the DNA there. Sperm needs to do nothing else. The sperm isn't going to go through mitosis or meiosis again. It's not going to do a lot of the normal cellular functions. So we start with the first cell, that's a diploid, and we go through meiosis. It ends up with 4 sperm cells. The process of the egg is a little bit different, a little more complex, because whereas the sperm only contributes DNA, the egg also contributes half the DNA, but then it also contributes the organelles. It contributes your mitochondria and all of those things. The only thing that the egg is lacking to become a full zygote, a full somatic cell, is it needs another half the DNA. That's what's going to come from the sperm. So whenever we go through meiosis, we actually end up with one mature egg. Even though one cell divides twice, 1 divided by 2 divided by 2 equals 1. It's because there's not enough stuff. There's not enough cytoplasm and organelles. You can't divide it up evenly. If you divided it all up evenly, you'd end up with 4 eggs, none of which would be mature enough to be able to form a zygote. The egg needs a lot of stuff. So 3 of them, 3 of the 4, are what we call polar bodies, meaning they're not mature eggs. They're going to end up being absorbed, broken down, and reused. Sometimes, if you look down the 2 tracks, the one on the left doesn't even bother going through meiosis 2. It just stays as a polar body. And so you end up with a medium-sized polar body, 1 small polar body, and 1 mature egg, as we see here. So 1 round of meiosis in the gametogenesis of sperm results in 4 sperm cells. 1 round of gametogenesis in eggs ends up in 1 egg cell. We have specialized names for it. We call it spermatogenesis and oogenesis. Okay, how about you pause and fill out a Venn diagram for me as they're developing B1 question for mitosis and meiosis. Well, mitosis only does 1 division, and meiosis does 2 divisions. Now, something they have in common is they both have interphase, prophase, anaphase, telophase. A difference is in meiosis, your homologous chromosomes pair up. They never do in mitosis. In anaphase 1 of mitosis, sister chromatids remain together, but in anaphase of meiosis, the sister chromatids separate and are ripped apart. Meiosis makes haploid cells and mitosis makes diploid cells. How about we do it again, this time for sperm versus egg? Okay, well, they're both haploids. They both come from the process of gametogenesis, which involves meiosis. Some differences is that the sperm is mainly DNA and motility, and that the egg is nearly a full cell, just lacking the other half of the genes. The egg has organelles that has a full set of organelles, whereas the sperm only has enough organelles to get it around. Another difference is that gametogenesis produces 4 sperm each time, whereas we only get 1 egg each time for the process. Proficient B3, are you equally related to your mother and your father? Justify your response. For all intents and purposes, yeah, pretty much. There is actually a slight edge towards being more related to your mother, though, since you get all the organelles from her egg. But you get half the DNA from each, so 23 chromosomes from each of them. So for all intents and purposes, yeah, half and half. But there's just that slight little bit. For example, we talked about mitochondria. You get mitochondria from your mother, and so you have no mitochondria from your father. Mitochondria reproduce themselves, so you have mitochondria like your mother. But for all intents and purposes, pretty much 50-50. All right, here's a tricky question to wrap your head around. If you were to travel in time, which common ancestor that you share with other life, other species, if you were to travel in time, which common ancestor would you have to go back in time farther to find? The common ancestor that all life that does mitosis shares, or the common ancestor that all life that does meiosis shares? Justify. Pause and think. Probably mitosis. Mitosis probably evolved longer ago, and meiosis was an advantage that developed as a change of it. Because we see parts of mitosis in meiosis, which means meiosis changed from that. We don't see parts of meiosis in mitosis. So mitosis is probably the older of the two.